Today I'm going to be talking about nucleic acids. So nucleic acids allow organisms to pass genetic information on to other generations. So they're exactly what you think they are. So they're DNA, RNA, tRNA, mRNA. So what does DNA do? DNA has genetic information, and you pass DNA on. So they're kind of the building blocks to organisms. So like DNA is kind of almost like the instruction manual to like how organisms are built before they're born. So nucleotides are like the smallest form of nucleic acids found. So they contain the following things: a phosphate group, which is here in the circle, a sugar, either deoxyribose or ribose, and a nitrogenous base, which is AT, GC, or U. So the phosphate group is pretty simple, but the sugar is either deoxy or ribose. So if you don't know this, get this now. Deoxyribose is DNA, deoxyribose nucleic acid. So ribose is RNA, and each of them have their specific base pairing. So nucleic acids are formed when several nucleotides bond together. So what happens is the sugar and the phosphate, let's go back to the other picture. So as you can see here, the sugar and the phosphate, they join together. So another sugar comes here and they join, and so does another phosphate. So they join and they create something which is called the sugar phosphate backbone. So it's kind of like it's the structure of the DNA. And the base here is left to bond with the other bases that are created in the other strand. So the history of DNA is pretty, I'm just going to be talking about Watson and Crick. So here you can see a picture of Watson and Crick. So they discovered the structure of DNA. So they discovered that DNA was a double helix. So it had two sugar phosphate backbones. And they got their ideas from a woman named Rosalind Franklin, who had some X-ray images that she didn't publish, but a friend of hers, she shared it with Watson and Crick. So soon after they released their thing, they got a Nobel Prize for it. So Watson, Crick, and also the other woman, they got a Nobel Prize. But Rosalind Franklin, she never got the Nobel Prize because she died before she even got nominated. So that's why a lot of people consider her to be the unsung hero of the history of DNA. We're going to talk about the bases of DNA and RNA. So the bases of DNA are adenine, which is A, thymine, which is T, cytosine, which is C, and guanine, which is G. The RNA bases are adenine A, cytosine C, guanine G, and instead of thymine, uracil. So you can see in this picture, RNA, instead of the thymine, they have a uracil. Oh, also, let me just quickly talk about the base pairings. So in DNA, A matches with T and C matches with G, while in RNA, A matches with U and C matches with G. So the structure of DNA and RNA are really, well, they aren't really different, but yeah, they are different. So here I have a Venn diagram. So as I said, the sugar phosphate backbone is the backbone of both DNA and RNA. So DNA has the bases ATGC, and they have deoxyribose sugar, which is why they have the name deoxyribose nucleic acid, and they're double-stranded. So they have two sugar phosphate backbones, so double helix. RNA, they have the base UTGC, and they have ribose sugar instead of deoxyribose, and they're single-stranded. So similarities between them are both of them have a sugar phosphate backbone, as I said. Like the number of them are different, but both are nucleic acids and both are made of nucleotides. So now I'm going to talk about DNA in crime scenes. So how DNA is used to solve many mysteries and crimes.
So we're gonna have a simulation of how we can use DNA to solve crimes. So it's pretty easy because if you have a DNA sample from each of the suspected criminals and a DNA sample from the crime site itself, you can figure out who committed the crime. So for example, here we have four culprits and we have their DNA. So the real DNA will not look like this, but it looks something like this. I mean the code, it's the code that we're looking for. So imagine the code of person one. Triangle, circle, circles, circle, circle. That's the DNA code of person one. Person two is circle, circle, triangle, square. Person three is square, square, triangle, circle. 4 is 4 is square, square, triangle, triangle. So we have the 4 DNA codes from each of the suspects. So now we need to look for the DNA that we found in the crime site. So for example, if the DNA we found in the crime site was circle, circle, triangle, square, it's pretty easy to find out who the criminal is. So let's see. So where do we have a circle in the beginning? So number 2. So do we have a circle in the beginning anywhere else? No. So let's just check the rest of the code so we're sure. So we have an other circle, triangle, triangle, and a square and a square. So this means that the culprit is this person because their DNAs match up.